This is the last module and lecture for exam 4. So once you complete this, you will have completed all of the material for exam 4. So this module is on development and inheritance. And so we're going to talk about development first, and then we'll move on to a little bit of inheritance. So the term development means the gradual modification of the anatomy and physiology of an organism from fertilization to maturity, right? So if you think about your development, you have been developing anatomically from the time sperm fertilized the oocyte all the way to your physical maturity. Now, if you remember back to the skeleton, you know that there are parts of your skeleton that are not physically mature. All the bones have not fused until you're about 25 years old. So that is your development for a human can continue up to about 25 in regards to anatomy. Physiology, you're essentially physiologically mature when you get to puberty, sometime between the age of 12 and by the time someone ends puberty and they're considered physiologically mature. Now, differentiation is the term that refers to what cells do in order for you to develop it. So these are different types of cells that are required for you to develop. So how does one cell become another type of cell in regard to function? So let's just stop for a minute and think about cloning. With the theory of cloning, you know that you should essentially be able to take one cell from a person, from anywhere in their body, and make another one of that person with all the functions, all the anatomy, all the physiology. Well, how does that happen? Because if you take a cell off your arm, a skin cell, how can you make another human out of that and have the liver function that you need to have, and the thymus function, or the ovarian or testicular function? How can you do that from a skin cell? Well, we know that yes, every single cell does have all of the contents that are necessary to make one of you, but some genes are turned off in each cell and others are turned on. So if you turn on a cell at a specific point in development that now says, okay, cell, instead of just being a generic cell, you're going to be a skin cell. Well, all the cells that proceed from that cell line from that point on are going to be skin cells. Fertilization is also known as conception, and this is when development begins. And we're going to go into great detail about what fertilization is and when it occurs. Looking at the stages of development, we see that there are 4: embryological, fetal, prenatal, and then postnatal. So embryological, everything that occurs the first 2 months after the oocyte is fertilized. If you study this, it's called embryology. Fetal development begins at the start of the ninth week and continues to birth. Everything that occurs from fertilization to birth is called prenatal development. That's the embryological and fetal development stages. And postnatal development is everything that occurs after birth, right? It continues to maturity. Once you hit maturity, the aging process begins. So that's something else you may not have ever considered. Once your bones are, for example, if we use your skeleton as an example, once you've fused everything, your sacrum's fused, your sternum's fused, then you actually begin to age, right? So you are aging. Once you hit 30, you begin to age. So this is my most favorite image out of all the images between AMP1 and AMP2. And I'll tell you why, because that single picture, the single image right here, tells such a huge story. So as you can see, it is sperm fertilizing an oocyte. So let's go back to how many sperm does it take to fertilize an egg? Well, clearly it takes thousands. You cannot say it takes one to fertilize an egg. Going back to what I've mentioned before, if that were true, that it only takes one sperm to fertilize an egg, then why would a low sperm count be an issue? And we're going to talk about what it means and why we need to have multiple sperm to fertilize an egg. So the term fertilization means the fusion of two haploid gametes. That means you have an oocyte that has the correct amount of DNA, the haploid amount of DNA, and a sperm that has the correct amount of DNA, the haploid amount. So when the oocyte and the sperm come together, they form a zygote, and it contains 46 unreplicated chromosomes. If you go back to what I talked about in spermatogenesis and oogenesis, and if we start with a spermatogonium or an oogonium that has 12 pounds of DNA, when it comes down to fertilization, the sperm will have 3 pounds and the oocyte will have 3 pounds. So when the zygote is produced, then how many pounds of DNA is it going to have? Well, it's going to have 6. Let's convert that to chromosomes. It has 6 pounds of DNA, but 46 chromosomes, but they're unreplicated. So once this zygote goes through, once the formation of the zygote occurs, then the DNA goes through synthesis. We still have 46 chromosomes. They're now replicated, and now we have our 12 pounds of DNA. Remember, 12 pounds is an extreme number, but it's something I want to have stuck in your head when you think about what happens during meiosis and what occurs during fertilization. Okay, so within a uterine tube, fallopian tube, within 24 hours after ovulation, that secondary oocyte is going to travel a few centimeters, and the sperm has got to cover the distance between the vagina and the ampulla of the fallopian tube. In order for the sperm to make its way into the oocyte, an enzyme called hyaluronidase is used, and it breaks the bonds with the follicular cells. 1, when it's still within the testes, and 2, it allows the sperm to actually reach the oocyte. So there is another enzyme that's called acrosin, and it's a proteolytic enzyme, and that too is required to reach the oocyte. The acrosomal cap of the sperm release both hyaluronidase and acrosin, and those are necessary to penetrate the corona radiata. So as you can see in this image, we have the corona radiata that I'm circling around right here, and multiple sperm are trying to get through this corona radiata, which is almost like a shell. So multiple sperm create the gaps in the corona radiata, and then you have a single sperm that makes contact with the oocyte membrane. When that happens, fusion occurs, and it triggers the oocyte activation, and it completes meiosis too. And this allows for the formation of the second polar body that you can see right here. So that is caused by oocyte activation. So as I said, this oocyte activation occurs because there is the fusion of the sperm cell membrane and the oocyte. And when this occurs, it allows for fertilization. Once that oocyte completes meiosis II, you can actually call it an ovum or an egg. So you notice that we had multiple sperm trying to fertilize that oocyte. Well, the term polyspermy means that fertilization has occurred by more than one sperm. That is prevented, and it is a very rare occurrence. And if it does occur, the majority of the time, this event would not end up in a pregnancy. So why does this not occur? Well, because there is a cortical reaction. It's in the cortex of that oocyte/ovum. An enzyme is released that hardens the zona pellucida. So when you think about the oocyte/ovum, truly think about it like it's a shell. In order for that sperm to fuse, in order for it to come in, it has to get through the zona pellucida. But if you think about it like a shell, it softens in a spot, or a window opens, if you want to think of it that way. One sperm enters, and then once that enters, this cortical reaction occurs that slams the window or hardens the shell again. So once we have fertilization, a female pronucleus, here's the female pronucleus, and the male pronucleus will be formed. And this is the nuclear material that was found. So this was the 3 pounds and the 3 pounds, right? Together, because each one of these had 23 chromosomes, they, once you add them, will make the 46 chromosomes that I referred to earlier, right? So the pronuclei very simply contain the DNA that was in either the sperm or the oocyte. So when those separate pronuclei fuse, that is called amphimixis. And amphimixis is known as the moment of conception. So you can see the fusion in this image right here, and then not only the fusion, but we've moved along to metaphase of the first cleavage division. But once that fusion occurs, you have a zygote with 46 chromosomes, and fertilization is considered to be complete. All right, so amphimixis is the moment of conception, and fertilization is complete. Okay, so I referred to differentiation earlier. Changes in genetic activity of some cells, but not in others. So some cells will be triggered to have activity, others will not. When this activity occurs, it's called induction, as you have chemical substances that are going to affect the differentiation of embryonic cells, and it is a very, very complex process. We're not going to go into great detail here, but that is the name of the process, induction. The term gestation just means the time that is spent in prenatal development. So 9 months, 3 integrated trimesters. So now that we have had the fusion of the pronuclei and fertilization is complete, we've talked about differentiation, how different cells are going to have different jobs. Before this can occur, we have something that is called cleavage. So this is the cell divisions that occur to produce daughter cells. So the zygote is cleaved so that you have different daughter cells. It ends when a structure called a blastocyst contacts the uterine wall, and we'll talk about that more in just a minute. Once that blastocyst hits that uterine wall and has contact, then we have the process of implantation that occurs. So that's the attachment of the blastocyst to the endometrium. And once implantation occurs, then we can have these embryonic structures that are formed.